My name is Christina, and I'm a bioinformatician at DIPLOMICS, and I work on genome assemblies. If you've ever opened a genome assembly paper, seen a snail plot, and immediately thought, what am I looking at? Then this video is for you. A snail plot is a compact visual summary of genome assembly quality, capturing both completeness and contiguity. Completeness reflects whether the expected genomic content is present, while contiguity describes how fragmented or continuous your assembly is. In addition to the radial distribution, the plot includes several summary metrics, such as the assembly statistics in the top left corner, BASCO completeness in the top right corner, AT and GC composition in the bottom right corner, and then the genome or assembly size and the size of the largest contig or scaffold on the bottom left. Because it integrates multiple metrics in a single view, it can be quite dense at first glance, so I'm going to break it down for you step by step. Imagine taking all of the contigs or scaffolds of your assembly, ordering them by length, and then wrapping them into a circle. So the point at the top is where the two ends meet. This is your 0% starting point. So from there you move clockwise around the circle, and this represents the cumulative proportion of your genome. So as you move along the circle, you're effectively walking through your genome from 0% to 100%. The vertical line from the top to the center of the circle represents the size threshold axis, and this shows the scale of the contig or scaffold lengths. So in this example, it is ranging from 100 kilobases to 100 megabases. Now, if we add the composition layer, the outer ring shows the GC and the AT content across the genome, shown in the two shades of blue. This can give you a quick indication of the base composition consistency. For eukaryotic genomes, you would expect this to be relatively uniform, so large fluctuations can indicate biases or contamination, for example. Next, we add the contig and the scaffold distribution, shown in gray. Here, all contigs are stacked by length, creating a cumulative view of the genome contiguity. To interpret this, you look at how far the gray region extends towards the center of the circle, and at any point you can read the minimum contig size on the radial axis, as well as the proportion of the genome made up of contigs of at least that size. Next, we have the longest scaffold or contig, which can be read on the radial axis and the center of the circle, which corresponds to around 100 megabases. And the red shading shows that this single contig makes up about 8% of the entire genome. Next, we have the layer that shows the N50. By now, you know that the N50 means that 50% of the genome is made up of contigs of this size or larger. To interpret it, you move clockwise around the circle towards the percentage of interest, you move towards the center of the circle, and you follow the line to the radial axis, and you read the size there, which corresponds to roughly around 20 megabases. Next, we have the layer that shows the N90 in the light orange on top of the N50. N90 means that 90% of the genome is made up of contigs of the size or larger. To interpret it, you move clockwise around the circle to the percentage of interest, move towards the center of the circle, move around and read the value of the radial axis, which corresponds to roughly 5 megabases. And if you were wondering why it's called a snail plot, it's because of the spiral shape in the center of the circle. It resembles a snail shell. The gray spiral represents the log-scaled scaffold or contig count, and the white rings increase in powers of 10, like 10, 100, 1,000, 10,000. And the further the spiral extends outwards, the more scaffolds there are. So to interpret it, for example, if you have 200 contigs, the gray spiral would extend past the 100 ring, but won't reach the 1,000 ring yet. If you have around 4,000 contigs, the spiral would extend past the 1,000 ring, but it won't reach the 10,000 ring yet. And if you have around 12,000 contigs, the spiral would extend past the 10,000 ring. And if you have less than 100 contigs, you won't see a white ring. So what does a good snail plot look like? A high-quality genome assembly would have a gray distribution extending deep towards the center of the circle, you would have high N50 and N90 values. Your BOSCO would be close to 100% with a low duplication percentage, and you would have a low contig count. What does a poor assembly look like? Your grade distribution would be around the edges of the circle, your N50 and N90 values would be low, your BOSCO completeness would also be low with high fragmented or missing percentages, and you would have a high contig count. So instead of looking at just the individual values of your genome assembly quality, the snail plot gives you an holistic view of all these values in one single glance. So just to note, this is my own interpretation of the snail plot, so if you have any comments or suggestions, please add it to the comments below.